Okay, so I'm going to talk about work that Trader, who's just here, has been doing. So Trader's a PhD student. He's getting into the last end of his PhD, and he's been tackling a problem that gets to this idea. So what we want to know is how drugs work. So it's a simple thing. In principle, all we've got to do is we've got some hole in our protein, and we've got a drug which is some shape, and the drug binds to the protein, and maybe it blocks this site from other drugs getting into the site, or it makes the protein change shape and acts as a switch in some way. And really this is the way that lots of people think about drug binding. And we'd like to get a more I guess realistic view of how this process might work. What we do is, so maybe I should introduce myself a little before we go. So I'm David Chalmers. I'm at the major piece of Monash that's located an hour or so back towards the city. We're essentially the pharmacy school. We teach 2 degrees. The largest degree is pharmacy students, but we also have a substantial pharmaceutical science degree, which is essentially a chemistry degree with some biology and pharmacology thrown into that. And we have research within the faculty which is really all aimed at some aspect of drugs. So how do drugs work? Can we make new drugs? Can we deliver drugs? So can we make an appropriate medicine to get them delivered into the body in the right way? So all of those aspects. I work more at the drug development end of the process, so finding new things that might one day be drugs or starting points for drugs. And in the drug development process, what we're interested in is we have some molecule. Generally, we want to know this thing. We want to know the binding affinity. How tightly does my molecule stick to my protein? And anything we can do, this taking a molecule and improving the affinity is a long, slow and expensive process, and anything we can do to improve our understanding of this process is useful. So I have my fingers in a few pies, but one of them is molecular dynamics. So what we want to know is, what we would want to be able to do is we want to build a 3-dimensional model of the drug binding to the protein with a realistic model. So we're going to model all of the atoms in the protein, or nearly all of the atoms in the drug. And the way we do this is we use a classical mechanics physics model. We can do quantum mechanics calculations, but the systems we're interested in have tens of thousands to maybe even millions of atoms, and the problem is too big to use quantum mechanics. So we need to use a classical mechanics model. The models of the molecules are really pretty simple. The atoms are treated as attractive spheres, so that's the van der Waals interaction. Bonds are modelled as simple or slightly less simple springs. We want to model everything moving, so what we need to do is solve Newton's equations of motion for that, and we do this on systems that maybe have 20 to 100,000 particles, so each particle is an atom in the system. So we made a lot of approximations in here, but these simple molecular mechanics models or classical mechanics models work pretty well for lots of systems, provided we're not interested in the quantum mechanical properties of the system. So we can't do anything that involves proper quantum mechanics. So for something like a drug binding, we can pretty much ignore the quantum mechanics and get a reasonable answer, and we get what we think are reasonable approximations of what's going on. Now there's no guarantee, it may work. Let's look at this. Aha, wonders will never cease. So here's a molecular dynamics model of a drug. So this is the drug haloperidol here. This is a protein. This is a G protein coupled receptor, which is a protein that's responsible for transferring signals across a cell. So what I haven't shown here is the cell membrane that sits along there. So this side is the outside of the cell. This side is the inside of the cell. And the job of these proteins is to somehow transfer a signal from this molecule binding in to a pocket on the outside through, and it produces a change in behaviour on the inside, and then that's recognised by other proteins on the inside. So this is a signal transducer. It takes the signal from the outside and it transfers it to the inside of the cell. And this binding process here is what we want to understand. So we want to understand how the drug, this is how it finally fits in the protein, and people have spent a lot of time looking at that. What they've spent less time doing is working out this process that I just showed the animation of. So it's actually a simulation, it's not an animation, of the drug moving from the exterior into the interior binding pocket, how it gets there, and we think that if we can understand that process better, there may be ways that we can interfere with this process or influence the process, or maybe design the process to go more quickly or more slowly, or maybe to have selectivity between this type of G protein coupled receptor and another one. So what we want to understand this process better, and to do that we can run simulations like the one that I just showed. That's a 200 nanosecond simulation. The big problem is that these processes this was an unbiased simulation, so we just put the drug out here and let it wander and find its way in. But we ran a set of 35 odd of these simulations, and only because it was unbiased, only in 2 of them did that binding event occur. And the other thing is it took Trader a couple of years to do all of this using high performance computing. So this is not an efficient way to go about doing these sorts of studies. So here's our problem. Our molecular dynamics simulation that I showed you, so we're simulating the progress of all the atoms in this 10 or 20,000 atom simulation. The size of so we calculate all the forces, move everything a bit, calculate all the forces, move everything a bit as we go. We're limited by the system to a short time step of about 1 femtosecond. So that's 10 to the minus 15th of a second. We can typically do 100 nanoseconds per day of simulation time on a high performance computer, but our binding occurs on a time scale that happens in a factor of 10 to 10,000 times what we can do in a day. So we can't, if we use an unbiased simulation, we need somewhere between 10 and 10,000 days of computer time to do that, and it's not an efficient way to do this. So Trader, after bashing his head against the table for some time, said this is no good. He went and read the literature, and one of the developing methods to do this is a thing called Markov state models, which was really brought to the fore by this group led by Pandey, and what they've used it for is looking at how proteins fold. So proteins are chains of amino acids that are folded up. They set up this thing called folding at home, where they break the simulation down into lots of little bits and they feed it off to people's desktop computers to run as a screensaver, and then they feed them all of the results back. And so they look at this, taking this extended chain up and to forming a folded protein. So it's been used quite a bit for this protein folding problem, but it hasn't been used much to look at the drug binding problem that we're interested in. So we're interested in using this Markov state model method. How much longer have I got? 4 minutes. To look at drug binding instead of protein folding. So what do? We take somewhere between 300 to 1,000 individual molecular dynamics simulations. And for each of those simulations, we've got 4,000 snapshots of the simulation as it's gone through. So we've got a big data set. It's too big to deal with, so we need to do some sort of reduction of the dimensionality to get it to squash down to look at the bits we're interested in. And then what we want to do is we can imagine our ligand binding. So if this is the energy of the system, it's going downhill in energy, and it's maybe got some low energy wells along the way. So we're going to call these different arrangements, different states, and we want to work out all of these individual probability distributions that we're going to find. So if this is lower in energy, we've got a higher probability it will be down there, and we want to know the distribution for all of these individual pieces, and then we want to reassemble that to give us an overview of the whole process. So we need to cluster our number of states. We need to count how many times it transfers backwards and forwards between these two energy levels, which depends on the energy difference. And then we want to build some network which has the probabilities of all the transitions in. And this is describing our binding process. And then we can go back and we can look at exactly what all of these individual states are, work out how important or relevant they are, and then work back to understand what that might mean about the overall binding. So this is a model that Trader built of the same binding problem that I just showed you the simulation of, and he's tried to pull out the major states, and he's tried to get a probability of the transitions for all of the network. What we have here is something that we think is kind of qualitatively right. We don't think it's quantitatively correct. So this is what gets us to our problem. We're interested in ways that we can improve this process. So we're interested in, I'm not a mathematician, I'm interested in things that would help any of these problems. Are there better ways we can build our transition network? When we've built one, how do we assess what the quality of the network is? Answer this question, is the system undersampled? Do we have too few samples? Almost certainly yes. So we want to know to what level, whereabouts is it undersampled. If we know that, we can go back and maybe do some more sampling and improve our model. We've done this as an iterative process. We've run some simulations and then tried to work out where we're undersampled and run more. Can we do it using a hidden Markov model? What are our best methods for data reduction, and what are the best methods for clustering the data sets pretty big? And can we sensibly, if we do use different methods, can we compare different approaches that we've used to generate our model and work out the best way to do it?